Scientists had a rule. Certain viruses were dead ends in humans. You could catch them, maybe die, but you could not pass them to the person next to you. The chain stopped. That was settled science. Then 4 different viruses, on 4 different continents, proved that rule catastrophically wrong. NIPAH virus, 75% fatality rate, clearly spreading between people in Bangladesh hospitals. Marburg, Ebola's close cousin, spreading in a cave in Uganda that tourists visit. Ebola, the one that kills 90%, appearing in a Virginia suburb and somehow infecting humans without killing a single one. And Lujo, a virus nobody had ever seen before, spreading hospital to hospital across 2 countries before anyone knew what it was. And here's the question tying all 4 of them together, the one that virologists consider the most consequential open question in infectious disease science. How many more viruses are out there, right now, silently capable of doing this, and we just haven't seen it happen yet? By the end of this video, you'll know exactly how viruses acquire this capability, which ones are closest to the edge, and why scientists are watching one specific pathogen more nervously than any other in 2026. Let's go. First, important framing. When I say a virus learns to spread between people, I'm not saying it's plotting anything. Viruses aren't alive in the traditional sense. No brain, no goals. But here's the thing, they don't need any of that, because what they do instead is simultaneously simpler and more terrifying. Every time a virus replicates inside a host, there's a chance of a copying error, a mutation. Most mutations do nothing useful. Some weaken the virus, but occasionally, one mutation or a combination makes the virus better at something, like infecting a new type of cell, or spreading to a new host species, or spreading between members of a host species that previously couldn't pass it to each other. That's it. That's the whole mechanism. Simple and terrifying. Here's a key piece of biology. The viruses that tend to do this rule-breaking, NIPAH, Marburg, Ebola, Lujo, the Andes hantavirus, are all RNA viruses, and RNA viruses mutate at roughly 1 million times the rate of DNA viruses. The reason? RNA replication uses an enzyme with no proofreading function. Every copy is imperfect. Multiply that across millions of viral replications happening inside one infected person, you get an enormous amount of genetic variation happening constantly. Virologists talk about the spillover window, the period during which a virus has just made the jump from animal to human and is still, in evolutionary terms, exploring its new environment. This is the most dangerous phase. The virus isn't yet optimized for humans, but it's running experiments constantly with every replication. And all it needs is one good result. 4 viruses. 4 case studies in what happens when the transmission rulebook gets torn up. Let's start with the one scientists are most worried about right now. Malaysia, 1998. NIPAH River District, Perak State. Pig farmers started dying. Brain inflammation, seizures, coma, death. 105 people dead in total. The Malaysian government culled over 1 million pigs trying to contain it. 1 million pigs. And they still didn't fully understand what was happening. The natural reservoir for NIPAH? Giant fruit bats, specifically flying foxes, genus Pteropus. They carry the virus permanently, never get sick from it, shed it in urine, saliva, and half-eaten fruit. What happened in Malaysia was ecological. Deforestation and drought pushed the bats out of their forest habitat and toward human settlements, including pig farms. The bats dropped fruit, contaminated with bat saliva and urine, was eaten by pigs. The pigs amplified the virus. Pigs and humans share close contact on a farm. The rest is tragedy. What Nipah does inside you is particularly brutal. It's not primarily a lung destroyer like the Andes virus. It's a brain destroyer. Nipah causes viral encephalitis, direct infection of brain tissue. The virus crosses the blood-brain barrier, one of the body's most fortified defenses. Once inside the brain, it replicates there. Fever, then headache, then confusion, then seizures, then coma. Symptom onset to coma, sometimes days. Case fatality rates in different Nipah outbreaks have ranged from 40 to 75%, depending on strain and available medical care. There is no approved human vaccine. There is no approved antiviral. Treatment is supportive care and hope. Just like the Andes hantavirus, just like Marburg, just like Lujo. Are you noticing the pattern? The 1998 Malaysian outbreak showed limited person-to-person spread. Most got it from pigs. But subsequent Bangladesh outbreaks, going back to 2001, showed a very different picture. The Bangladesh strain clearly spreads between humans. Healthcare workers catching it from patients. Family caregivers catching it from family members they were nursing at home. In 2004, a chain. Person A infected B, who infected C, who infected D. Classic sustained human-to-human transmission. Confirmed. India got a very close look at Nipah in 2018. The Kerala state outbreak started when 3 members of one family died from an unidentified illness. The youngest, 23 years old, had likely been exposed to infected bat droppings from a date palm tree near his home. The virus spread to family members, then to healthcare workers. 23 people died. And then, something remarkable. Kerala mobilized 30,000 contact tracers, traced over 2,000 contacts, quarantined aggressively, stopped it in under 2 months. The WHO has Nipah on its R&D blueprint priority pathogen list, the short list of diseases most likely to cause an international health emergency and for which no adequate countermeasures exist yet. And what keeps virologists up at night specifically about Nipah? The fruit bat reservoir lives across a huge geographic range, from South and Southeast Asia all the way to parts of Australia, and climate change is expanding that range. Just like the Andes rat, just like the Egyptian russet bat with Marburg. Quick pause. Before we get into Marburg, drop it in the comments. Which of these 4 viruses do you find most concerning? Nipah, which already clearly spreads human to human. Marburg, Ebola's close and equally deadly cousin. Ebola Reston, the strain that somehow infected humans without killing them. Or Lujo, the one almost nobody's ever heard of. Let me know below. I'll reply to as many as I can. Okay, Marburg. Marburg is one of the oldest known viral hemorrhagic fevers, and it has an unsettling origin story. In 1967, in Marburg, Germany, and simultaneously in Frankfurt and in Belgrade, laboratory workers who had been processing African green monkeys imported from Uganda started getting sick and dying. 31 people infected, 7 deaths. The monkeys were being used for polio vaccine research. They came from Uganda carrying a virus nobody had ever encountered. Marburg belongs to the Filoviridae family, the same family as Ebola. Under an electron microscope, they look terrifyingly similar. Long, filamentous, thread-like particles, like tiny biological whips. It was the first filovirus ever discovered, predating the identification of Ebola by 9 years, and it remains one of the most lethal pathogens ever documented. Marburg is a hemorrhagic fever. It attacks the lining of blood vessels throughout your entire body. Early symptoms look like a bad flu. Then the gastrointestinal phase, violent vomiting, severe diarrhea, massive fluid and electrolyte loss. Then, in severe cases, the hemorrhagic phase, bleeding from multiple sites as vessel walls fail, liver failure, kidney failure, multi-organ collapse. Case fatality ranges from 24% to 88%. The 2004 to 2005 Angola outbreak hit 88%, the highest ever recorded. Marburg spreads between humans through direct contact with blood, secretions, and organs of infected people, and through contaminated surfaces. In the 2004 to 2005 Angola outbreak, the virus spread aggressively within healthcare facilities because workers lacked protective equipment and the outbreak wasn't identified as Marburg fast enough. By the time anyone knew what they were dealing with, the hospital itself had become a transmission engine. One of the most dramatic Marburg transmission stories involves a cave, the Python Cave in western Uganda. A popular tourist destination, visitors went in to observe massive colonies of Egyptian russet fruit bats on the ceiling. In 2008, a Dutch tourist visited, died in the Netherlands shortly after. An American tourist visited the same cave, died in Colorado. Both caught Marburg from the bats, or more precisely, from bat droppings and urine contaminating the cave environment they walked through. The Egyptian russet bat, Russetus aegyptiacus, is the confirmed primary reservoir for Marburg. Just like the fruit bats with NIPAH, just like the pygmy rice rat with the Andes virus. The bat carries the virus permanently, sheds it in droppings and urine, and is completely asymptomatic. These bats live in colonies that can number in the millions, spread across sub-Saharan Africa. Every colony is a potential reservoir. Every cave is a potential exposure point. As recently as 2023, Equatorial Guinea experienced its first ever documented Marburg virus outbreak. 9 confirmed deaths, 15 probable cases, linked to bat exposure in forested areas of Kiantem province. Person-to-person spread within family and healthcare settings confirmed. The WHO mobilized, international response teams deployed. It was contained. But it was a reminder, Marburg is not a historical curiosity. It is active, distributed across sub-Saharan Africa, wherever its bat reservoir lives. And here's the detail I want to make sure lands. There is no approved Marburg vaccine for general human use. Several candidates are in development. Johnson & Johnson's candidate showed early clinical trial promise. But as of right now, if you're exposed to Marburg, the treatment is supportive care and hope. We've known about this virus since 1967. It's been killing people in documented outbreaks for nearly 60 years. And we still don't have a vaccine on the shelf. Okay, this one. This one is the entry on today's list that I think genuinely deserves to be more widely known, because it is, in my opinion, the most alarming near miss in the history of virology. 1989, Reston, Virginia, a quiet suburban town just outside Washington, D.C. A primate research facility received a shipment of 100 long-tailed macaques from the Philippines. The monkeys started dying at an unusual rate. Scientists were called in. Tests were run. Results came back. A filovirus. A member of the Ebola family. In a monkey facility. In a Virginia suburb. The CDC and the U.S. Army Medical Research Institute of Infectious Diseases moved in immediately. They euthanized all 450 monkeys in the facility, decontaminated everything, and then they discovered something that genuinely shocked them. Several human workers at the facility had seroconverted. Their blood showed antibodies to the Reston Ebola virus, proving they had been infected. All of them were completely asymptomatic. Not a single human got sick. Now, that sounds like a relief, and it was. But here's what made virologists genuinely stop breathing for a moment. Here's the part that keeps virologists awake. The Reston virus appeared to spread between monkey rooms that had no direct animal contact with each other. Isolated rooms. The most plausible explanation for how the virus traveled between them? The air handling system. Airborne, or at minimum droplet, transmission between non-human primates. Ebola. Airborne, between rooms, in Virginia. The story got more complicated in 2008. Reston Ebola virus was detected in pigs in the Philippines, the first time any Ebola virus had ever been found in a non-primate domestic animal. Philippine pig farm workers tested positive for antibodies, again, all asymptomatic. And pigs are a known influenza mixing vessel. They can be infected by both avian and human flu strains and serve as an evolutionary mixing chamber. The question: could pigs do the same thing for Reston Ebola virus? The lesson of Ebola Reston is not, sometimes Ebola is harmless. The lesson is, a filovirus with apparent airborne transmission capability between primates has already successfully infected multiple humans without being detected immediately. And the only reason it wasn't catastrophic is because this strain happened to be non-lethal in people. That combination of features, spread capability plus current low human pathogenicity, is exactly the kind of silent progression scientists most fear. Because by the time pathogenicity increases, the spread infrastructure may already be in place. The Reston incident is the event that inspired Richard Preston's book, The Hot Zone, published in 1994, which put this near miss into public consciousness. It became a massive bestseller. Then, a National Geographic television series in 2019 starring Juliana Margulies. So millions of people have heard a dramatized version of this story, but far fewer know the actual documented science behind the event. The real story is more complicated and more unsettling than the dramatization. Lujo virus. I guarantee most of you have never heard this name, and that's the point. In 2008, a cluster of cases emerged across Zambia and South Africa. The name is a portmanteau: Lusaka, Zambia, and Johannesburg, South Africa, the two cities at the center of the outbreak. 5 people total infected, 4 died. That is an 80% case fatality rate from a virus nobody had ever seen before, that appeared, killed, and vanished in a matter of weeks. The index patient was a woman who worked as a safari camp manager in Zambia, living and working at the edge of wild habitat, the exact human-wildlife interface we've been talking about. She fell seriously ill and was medevaced to a hospital in Johannesburg for treatment. During that treatment, she was already contagious. And the people who cared for her and the paramedic who transported her, they didn't know what she had. Nobody did. It was brand new to science. Over the following days, 4 healthcare workers and 1 paramedic who had been involved in the index patient's care became ill. The index patient died. 3 of the 4 secondary cases died. 1 survived. The person who received an experimental ribavirin treatment early enough in the disease course. 4 out of 5 people dead, in a hospital, in a major city, from a virus science had never cataloged before that week. The Lujo outbreak demonstrates something that connects every virus on today's list: the identification gap. The time between when a novel pathogen first starts infecting healthcare workers and when it's identified as something requiring full hemorrhagic fever precautions. Healthcare workers were using standard protections, appropriate for flu or routine infectious disease, not the full PPE needed for a known hemorrhagic fever. By the time Lujo was identified as a novel arenavirus through CDC advanced diagnostics, 3 healthcare workers were already dead. Here's what makes Lujo especially significant from an emerging disease standpoint. It appeared completely without warning. No prior documented cases. No known confirmed animal reservoir, though a rodent is strongly suspected, consistent with other arenaviruses. A total of 5 human cases and 4 deaths before it disappeared as quickly as it arrived. We don't know how many undetected cases occurred. We don't know if people in rural Zambia have antibodies from past silent exposures. In a very real sense, Lujo is still a largely closed book. The single Lujo survivor received ribavirin, an antiviral used against other arenaviruses like Lassa fever. early enough in the disease course that it likely made a critical difference. The other patients did not receive it in time. This is a recurring theme across these viruses: the narrow therapeutic window, the small gap between early enough to help and too late. And the only way to hit that window is to identify the pathogen fast enough to know which treatment to reach for. Which brings us back to the identification gap. Here's where the story shifts from what's happened to what we're doing about it. USAID ran a program called PREDICT from 2009 to 2020, specifically aimed at surveying animal populations globally for novel viruses before they spill over into humans. The program sampled hundreds of thousands of animals across 31 countries. Results: over 900 novel viruses identified with pandemic potential. 900. Previously unknown to science. Living in animal reservoirs right now. A landmark 2018 scientific paper estimated there are approximately 1.67 million unknown viruses living in mammal and bird populations globally, and roughly half could theoretically infect humans. We have formally identified maybe 0.1% of that total. The rest is, scientifically speaking, dark matter, unknown, unstudied, sitting in bat colonies, rodent burrows, bird populations all over the planet. Most will never infect a human in a meaningful way, but even a small fraction of 1.67 million is a very large number. The single most consistent driver behind every virus on today's list, and behind COVID-19, and behind HIV, and behind influenza, is the human-wildlife interface. The boundary between natural ecosystems and human activity. Every time forests are cleared for agriculture, every time wildlife habitat shrinks, every time a new road cuts through previously remote jungle, that boundary gets thinner. Animals that previously lived nowhere near humans now live next to farms, next to markets, next to cities. And the viruses they carry come with them. Virologists evaluating pandemic risk assess 4 key factors. 1. Can the virus bind to human cell receptors? 2. Does it replicate effectively in human respiratory tissue, the most efficient transmission route? 3. Can it survive in the environment long enough to reach a new host? 4. How fast does it mutate, and in how large a host population, determining how many evolutionary experiments it runs. RNA viruses in massive animal reservoirs with high mutation rates check every single box. Which is why bats, which live in colonies of millions, fly long distances, and carry RNA viruses with extreme mutation rates, dominate this risk landscape. Here's the genuinely hopeful part, and I want to make sure this doesn't end on pure dread. The tools for identifying novel pathogens in 2026 are incomparably better than even 10 years ago. Genomic sequencing that took months in 1967 and weeks in 1993 now takes 24 to 48 hours. Metagenomic surveillance, the ability to screen samples for viruses present, not just ones we're specifically hunting, is rapidly improving. The COVID-19 pandemic was devastating, but it produced massive investment in pandemic preparedness infrastructure and mRNA vaccine technology that can be adapted to new targets within weeks. Here's where I want to land this. Scientists aren't wrong when they say a virus doesn't typically spread between humans. That statement is usually true. But usually in virology needs constant vigilance behind it, because the Andes virus usually didn't spread person to person, until it did. Nipah usually came from bats to pigs to humans and stopped, until Bangladesh. Ebola usually didn't spread through air, and Reston may still not in people, but it did between primates in a Virginia building. The rulebook isn't wrong. It's just incomplete, and these viruses are the annotations in the margins. Every single virus on today's list shares one terrifying commonality. It was already spreading between people before anyone knew what it was. Marburg workers were dying before the pathogen was named. Lujo healthcare workers were infected before the virus existed in any database. The Andes virus had already created a superspreader chain before the transmission mechanism was characterized. The identification gap is the most dangerous interval in outbreak science, and shortening it is the single highest leverage investment global health can make. But here's what I want the takeaway to actually be, because it's not just dread, and it shouldn't be. The Kerala NIPAH response in 2018 was contained by 30,000 contact tracers and decisive, rapid public health action. That works. Outbreak response, when properly resourced and rapidly deployed, genuinely saves lives. Marburg outbreaks have been contained repeatedly. The Lujo chain was broken. The Ebola Andes virus outbreak was stopped. The tools exist. The question is always whether the political will and the resources exist to deploy them at the speed these pathogens demand. NIPAH, Marburg, Ebola, Reston, Lujo. 4 case studies in viruses that broke, or partially broke, the rules we thought governed their transmission. 4 reminders that the line between animal disease and human outbreak is thinner than most people realize. The next novel pathogen capable of human-to-human spread isn't hypothetical. Given what we know about spillover rates, viral mutation rates, and the expanding human-wildlife interface, it is a matter of when, not if. What determines whether that when becomes a small, contained outbreak or something much larger is the speed and quality of the response. And that response starts with people understanding what these viruses are and how they work. You now do, and that matters. Next episode, we're going deep on bats. Because if you've been paying attention, you've noticed they keep appearing in every single story we've told. NIPAH, Marburg, Ebola Reston, Lujo, probably. Bats are, per capita of species, the most virus-rich mammal group on Earth. And the biological reason why bats can carry dozens of lethal viruses without getting sick, while we get absolutely devastated by them, is one of the most fascinating stories in evolutionary biology. It involves their immune system, their metabolism, their extreme body temperature during flight, and it may hold the key to treating a range of human diseases. Subscribe so you don't miss it. And if today's video surprised you, share it. Most people have never heard any of these names. That needs to change.